Kelly, it will be live on YouTube.
Okay.
Just for your information. Sure. Okay. What is sir? What about other presenter?
She will join.
She is joined?
Yeah. She will join. Okay, we'll start.
Yeah.
Hello everyone. Good morning, good afternoon, or good evening, depending upon the participants from which part of the world you are watching this program. Thank you very much for joining with us today. On behalf of my institute, Central Institute of Brachycephalic Aquaculture, Chennai, India, I welcome you all to this event. I'm sure you all know about the antibiotics. After its discovery, it was considered as a wonder drug because it was able to save a lot of life. But if you see the present situation, it has come back like a boomerang. Instead of saving life, it is taking a lot of life. And who are responsible for this? It's we. All because of our irresponsible act, the situation has become very nasty. And you know, there are many factors for a healthy life, and awareness perhaps occupying the prime position. And maybe for this reason, the World Health Organization every year is organizing this awareness program and teaching us how to handle the antimicrobials in a more responsible manner. And as a small support to this program, our institute is organizing this awareness program, and we are sure that it will serve the purpose in educating many of the participants regarding the misuse of antimicrobials. So our institute has been working on this antimicrobial program, mainly the antimicrobial resistance, through a project called INFAR, where we have a coordinated research project between the ICR and FAO, and also we have a All India Network project on fish health, where we are working on antibiotic residue. So through these projects, we are organizing this awareness program. And today we have two speakers with us. One of our speakers is Dr. Kelly Thamban from University of Exeter, UK. So she's a very young and enthusiastic research. And for quite some time, she has been in the field. In this field, she has been doing the research, like AMR. And his work is mainly on aquaculture practice. So I'm sure if some of the farmers and the stakeholders, if they have joined us today, definitely they will be benefited by this. Our second speaker today is Dr. Anusha Rohit, who is from Madras Medical Mission, Chennai in India. And she is heading the microbiology department there, and she is also chair for the infection control. Again, she is a very young and talented medical practitioner as well as a researcher, and she has been speaking in many forums. And I'm sure as a whole, all the participants will get benefited through her talk. Now, before we go to the talk, now I will request my colleague, Dr. P.K. Patil, to say a few words regarding the importance of this event. Dr. Patil, please.
Yeah. Thank you, Dr. Otta. This was a nice introduction, and just for the benefit of our audience and key speakers, this institute is mostly concentrating on shrimp culture farming in India. So we are mostly interested in farming and the problems in shrimp farming, and how the practices, better management practices, and other activities in controlling infectious diseases will have an impact on antimicrobial usage. So as Dr. Otta said, annually, every year we conduct this program, World Antimicrobial Awareness Week 2020. Like this we conduct every year, and as a part of this program, we have already conducted drawing competition and a speech competition for our research scholars and students and children of our scientists they did a drawing competition, and at the end of this program, we will be announcing prizes for the best drawing, best speech, best quiz winners. That is only to generate interest in the young people as at student level. At the school level, now this issue is being addressed. Even our children, those who are in school also, they are aware, and they, when we take any medicine, they will ask us, is it an antibiotic or something else? Are you taking a full dose? That is an awareness which has been created throughout the world, and especially in India. So in this connection, now this antimicrobial awareness week is not only restricted to AMR, though our speakers will be talking more on AMR, we should be concentrating other areas also, like its usage pattern, and its withdrawal period, and then its dosage and all. As some of the things which we are very casually using some words like antimicrobials and antibiotics, because antibiotics are used for different purposes, used for therapeutic purpose, prophylactic purpose, and growth promotion purpose. Definitely, as we know, antibiotics used for therapeutic purpose have less impact on this AMR, and anyway, antibiotic used as a growth promotion or as a prophylactic purpose are only having a greater impact on AMR. And as we know, in India, we are doing a survey, and I am also a part of one program wherein we are doing a lot of survey on what is the usage pattern of aquatic drugs and medicines in aquaculture, including antibiotic. Definitely antibiotics included. So we are doing a national survey, and all we have seen among all the more than 3,500 different products being used in Indian aquaculture, hardly 4%, 2 to 4% of them are antibiotics, and that too being used in fish for therapeutic purpose only, not as a growth promoter or as a prophylactic purpose. So our usage pattern definitely is very less. So anyway, Kelly, she has worked in Bangladesh and different parts of the world. She can tell us what is the usage pattern in other countries. So compared to other developing countries, Indian usage pattern is definitely much less. So, but anyway, we have to be cautious, and we have to be aware of this problem. So with this introduction, this is our purpose. So next, I think we can call Kelly is the first speaker.
Hello.
Yeah. So thank you, Dr. Patil, for your brief introduction. Now I request Dr. Kelly to deliver her talk. Kelly, please.
Hello. Thank you very much. Thank you very much for the introduction and for inviting me to talk. I'm delighted to be able to share my work with you all and be part of this. I'll just share my screen.
Yeah.
Okay. And can you see that okay?
Okay.
Yeah, brilliant.
Yeah.
And you can see the full screen?
Yes.
Okay, yeah, it's not showing that on mine, so that's good. Okay, yeah, well, thank you very much for inviting me to talk. Yeah, I'm going to give you an overview of the work that I've been doing. I mainly work in Bangladesh aquaculture, so I can give you some experiences from that and talk about some of the projects we've been doing. So just to, first of all, is that not working? Oh, it's not changing. Let me see. Has the slide changed on your screen?
Yes, it's changed.
Oh, I can't.
What
okay.
I'm going to talk, yeah.
Okay, right. So does it say who am I?
Yes.
Okay, great. So so my name's Kelly Thornber. I have a background in medical research. I've worked in different types of medical topics, so cancer research, blood clotting, some infectious diseases, and I've also done quite a lot of science communication work. And currently I'm in Exeter, which is in the southwest of the UK, and the Biosciences building is somewhere in the middle there. And I work in the Centre for Sustainable Aquaculture Futures, which is a centre within Exeter, and we have links to the UK government centre. Oh, not changing. Oh, it's not changing again. Has it changed on your screen?
No.
Oh. Okay, is that better?
It's not yet changed.
It's not changed. Oh. Okay.
Yeah, now it's okay.
No, it's okay. Okay, brilliant. So yeah, what am I going to talk about today? I'm going to give a very brief introduction to AMR. I don't know how much everybody's background knowledge is, so I'll just go through it, and apologies if it's too basic. And then I'll talk about AMR and aquaculture, and the risks associated with aquaculture. And then I'm going to just go through 3 of the projects that we've been working on in Bangladesh, and then conclude. So going back to basics, if I'm going to talk about antimicrobial resistance, I think first of all it's important to clarify what are antimicrobials. So these are chemicals or drugs that kill microorganisms or microbes, and so they either kill them or they prevent their growth. And the most common antimicrobials I'm going to talk about are antibiotics, which kill bacteria. But just so that you're aware that antimicrobials includes antivirals, antifungals, antiparasitics, and even things like heavy metals, disinfectants, antiseptics, which are non-selective. So that means they don't target specific microbes, they sort of just kill everything. But they're still important for driving antimicrobial resistance, because when these chemicals are present in an environment, it makes the resistance to these other chemicals more likely to happen. And as Dr. Patil talked about just now, they're used to treat disease, prevent disease. So, for example, during surgery, medics often give patients antibiotics to make sure that, because they know they're going to have some bacteria possibly in the wound after surgery, so they give them antibiotics to prevent any infections growing, and they are used to promote growth. So in animals, even healthy animals, antibiotics have been shown to increase growth. Nowadays, this is banned in most countries, the use of antibiotics to promote growth, but it still goes on quite a lot. And as I know, I said before, antibiotics are really important for modern medicine. All these chemotherapy, prenatal care, treatment of major infectious diseases, surgery, this all really needs antibiotics and antimicrobials more generally. And we are going to be in a really bad situation when these really stop working, and they already are, as Dr. Otter said. Also, food security is really dependent upon effective antimicrobials. So with the growing global population, we have more intensive farming. So here's an intensive chicken farm, intensive aquaculture farm, where animals are in much closer proximity than they would be in nature. And that means that they're much more susceptible to infectious disease. And therefore, antibiotics are really important to allow us to continue farming animals in this way. So then, just to go over, what is antimicrobial resistance? Well, first of all, very basically, just to make everybody, I'm sure everyone's probably aware, bacteria and other microbes are absolutely everywhere, and they are fundamental to our Earth existing. They are responsible for, I think, around half of the production of oxygen in the atmosphere. They obviously decompose matter and release nutrients. They're essential for human health. The microbiome in our gut, all the bacteria and microbes in our gut, is really important for our health. And so, and what is happening though is, of all these microbes, when we add antimicrobial drugs to kill them off or to prevent their growth, they are developing resistance, which means they're sort of fighting back against the drugs. So the drugs are becoming less effective at treating them because they're getting better at fighting them. And some of these microbes they cause disease in humans and animals. And so what's happening is these microbes that cause disease, they are getting better at resisting the drugs. And so when they infect our bodies, they then don't cause, we can't treat them because the drugs don't work anymore. And the main factor driving antimicrobial resistance is antimicrobial usage. So here it's antibiotics. You can see if you've got a population of microbes, 2 of them might be naturally resistant, because it's a natural evolutionary process. When we add antibiotics, what we do is we kill off the bacteria that are called susceptible, and that means we are left with the ones that are resistant, and then they multiply and grow. So by using antibiotics, we are causing the growth of more resistant bacteria. And this is a particular problem because bacteria can actually pass the genes, that's the DNA that codes for resistance, they can pass it to each other, sort of like shaking hands with each other almost. So here, if we have these green bacteria, which are sort of friendly, if you like, they're not causing disease, but they have this squiggly, this is to represent the DNA that codes for resistance, and this is the red code, so they are resistant to antibiotics. And so if we have an environment where we have bacteria that aren't causing disease, but they are resistant to antibiotics, and into this environment we put some disease-causing bacteria in blue. So let's say this is an environment where some human pathogens, like outside a hospital, for example, in the wastewater treatment, it's full of environmental bacteria and human pathogens, which is disease-causing bacteria. So what can happen is this sort of friendly bacteria can pass the DNA to the human disease-causing bacteria, which means that this then becomes resistant and it then multiplies. And so what you end up with is an environment where you have not just, to human health, it doesn't matter if these green ones are resistant because they're not going to cause disease, but it really does matter if a human gets infected with one of these blue ones now, they're going to get disease that cannot be treated. And so that's why this is really important, and we need to stop the use of antimicrobials as much as we can. And then this was a report from, 6 years ago now, estimating the global burden of AMR. So if you look at, this was currently in 2014, globally there are around 700,000 deaths per year from AMR compared to all these other diseases and causes of death. Cancer, which is obviously a big cause of death, in 2014 was 8.2 million people per year, and the estimates are that by 2050, so in 30 years time, more people, around 10 million people per year, will die of AMR, and you think that's bigger than the number of people dying of cancer now. So it's going to become a bigger and bigger problem, with a huge estimated global cost of $100 trillion US. And the biggest problems are really going to be in lower middle income countries, particularly in Asia and Africa, where there is the sort of highest burden of infectious diseases, the lowest access to healthcare. So to go on to AMR and aquaculture. So what are the risks associated with aquaculture and AMR? Well, this is an example of a sort of smallholder farm, but I mean it relates to many aquaculture farms of a bigger scale. So around, as you probably know much better than I do, around 90% of aquaculture takes place in Asia, and in many countries in Asia there is a lack of wastewater treatment systems and sanitation infrastructure. And that means that this aquaculture farm, the water that it's receiving often is polluted by antimicrobials, heavy metals from industry, manufacturing, hospitals, all sorts of things upstream. And that might be also polluted by disease, so microbes that cause disease, and even resistant microbes. So the water that comes into aquaculture ponds often can be polluted already with antimicrobials or disease-causing microbes. And that makes it difficult right from the start for farmers, because if the water quality is poor, that affects the health of the animals. So it means they're more likely to need antimicrobials to keep their animals healthy. Obviously, they can practice better pond management to improve biosecurity, and that's something I'll talk about later. But we must remember that it's not easy for them either. The other thing is that on top of all whatever's coming in through the water, antimicrobials are heavily used in the animals and in the people who live at the ponds. And the antimicrobials come out in their feces. Often, I think about 80% of antimicrobials pass through unmetabolized. And so this all comes out and goes into the pond or into the local environment as well. In some farming systems, the sludge from ponds is used to fertilize the crops and feed the animals. And again, this is transferring potentially resistance and antimicrobial residues to around, and it's just making everybody more exposed to this. Also, the humans are more exposed to the water, particularly in these sort of systems where it's sort of extensive farming or small-scale farms. And so the humans have direct contact with these resistant microbes and the antimicrobial pollution. And then finally, the water gets released, again, often untreated. And so that goes downstream to the next farmer or the population living downstream. And then we also, from an international point of view, obviously, if you have animals that are reared in antimicrobial polluted water, they have antimicrobial residues, which then the consumers, then, when they eat them, they get exposed to those residues. And I know in Bangladesh, a lot of the shrimp industry, the shrimp is exported. So this is a way by which resistant pathogens, so resistant microbes in Asia can transfer across the globe because they get exported to Europe and the United States. But obviously this is also really important because for the local consumers, and I know for the fin fish market in Bangladesh, and that is what I think 90% of Bangladesh aquaculture is fin fish, and that predominantly goes for the domestic market or the global south countries. So it's a huge problem for consumers as well. Yeah, so as I said, for about 3 years now, I've been working, I switched from medical research and started applying my medical background to aquaculture. And I've mainly been working in Bangladesh. And in Bangladesh, aquaculture is a huge business, as I imagine it is in India as well. And over 17 million people, so 11% of the population, depend on aquaculture for their livelihoods. It provides over 60% of the animal protein, but there are high levels of disease, poor biosecurity, like I say, poor water quality, and generally poor access to diagnostics and advice. And that means that antibiotics are widely used, they're easily available, and it's very difficult to regulate the sales and usage with the resources that the government has, and the quality, obviously, and people generally have a low awareness of AMR. So, and Dr. Patil was asking about the levels of antibiotic usage in shrimp in other countries. I think it's great what you're doing in India, trying to gather information about this. From my knowledge, I don't think aquaculture usage of antimicrobials, from my knowledge, is quite poor. I think it's widely known that aquaculture usage in animal production in low and middle income countries generally, antibiotic usage in animal production generally in low and middle income countries is very difficult to monitor. But the additional problem in Bangladesh is that because shrimp are predominantly aimed at the export market, there are testing for animals that go to export. So the UK, well, Europe, the US test for antibiotic residues. So there's a lot of pressure on farmers to reduce their antibiotic usage. And that means that sometimes it can be difficult to get accurate data because it becomes a bit of a taboo subject that people don't want to talk about. So there's lots of challenges that I've found, and I'd be interested to hear if you find that also in India. So because of my background in science communication, I was really interested when I first started working in this topic on the levels of awareness and if we could increase awareness of AMR. And obviously in somewhere like Bangladesh, it's very difficult to raise awareness because awareness raising is labour intensive. These aquaculture farmers are usually very rural and difficult locations to get to, which makes it expensive as well. It takes up a lot of time to get around, lots of small groups of people at the time. And AMR is a complicated scientific concept to try and convey to people, especially those who maybe haven't had extensive education. And in Bangladesh, literacy has increased a lot in the last few years. At 2017, it was 73%, but it's still lower in rural areas. So there's a lot of challenges in raising awareness of AMR. And I had an idea to use digital communications because when I was visiting Bangladesh, I saw, even though these farmers are in the middle of nowhere, in very rural locations, there was really good 4G coverage and everybody had mobile phones, or it seemed. The coverage was better than some parts of the UK. I was absolutely amazed. And it was brilliant because that means that because there's so many mobile phone users and this is still increasing all the time, this was the hypothesis was that maybe we could use digital communications. And one of the advantages of this is that they can be very engaging and accessible, which means even people who can't necessarily read very well can still see the visuals. And obviously everybody loves watching very colourful, attractive media. So we decided we would try and develop a little animation, and at a workshop I went to, and I think it was February 2018 or 2019, I can't remember now, 2018 I think. I ran a session where with a lot of stakeholders from the aquaculture industry in Bangladesh, and we talked about what the important messages should be. And so from that we developed the animation, which I'm hopefully going to play now if it works. It's about 4.5 minutes long, so I'm just going to hopefully see it. If when I press play, please let me know if it doesn't work at your end.
Yeah, it works.
Can you hear it?
No sound, but can't hear anything, but the picture is coming only.
Oh, did you say you can't hear it?
Yeah, you can't hear.
You can't hear. Oh, okay. Okay, I'll skip it then, because if you can't hear it, you can't hear the messages. That's a shame. Okay, well, just you can watch it later if you want to at our website, but I think it is on YouTube somewhere as well. So the main messages that we identified were that, first of all, we need to make sure that we make it clear that we convey positive messages. So aquaculture can be very profitable, and is a good business, because we didn't want it to be too negative. So we start with very positive messages, and this is this down on the bottom left. This is where we try to explain visually how antimicrobial resistance works. These are in blue on the left. You see the antibiotics firing at the bacteria, and these ones with the white shield, you see the antibiotics bouncing off them. So this is trying to explain the concept of AMR. And then there were messages such as, make sure you mix your antibiotics with feed. Don't add the antibiotics directly to the pond. And don't use antibiotics in combination. So you should be using one antibiotic and not a mixture. Other messages were that if you practice good pond management, so here, for example, you can see a farmer building a fence around the pond to keep the animals out. This will help to reduce the disease and therefore reduce the need for antibiotics. This message on the top right, this was saying, if you do have disease and you think you need antibiotics, call your local fisheries officer for diagnostic advice rather than self-treating. And in the bottom right, the message is the fisheries officer will come and speak to you and write you a prescription to use the antibiotics. So these are the best practice messages that were identified by the stakeholder group as being the most important to get across to farmers. And we took this into the field. You can see that they showed it on a laptop, and the farmers seemed to really enjoy it. It was only a small pilot study to see if this would be effective, but from all the farmers surveyed, they all thought it was a good way of communicating with them, and 97% said it would change the way they use antibiotics in the future. Now, we didn't follow this up, so we don't know if this did change behaviour, but it supports the idea that digital animation can be used as an effective communication tool for conveying messages. Obviously, whether that supports behaviour change is a whole other study, which we will try and get to in the future. Excuse me. We did also release it on a small social media campaign, and in 2 weeks we had 20,000 hits through a Facebook group in Bangladesh, and 105,000 hits through Twitter outside Bangladesh. And again, this shows a lot of potential for digital communications that, imagine the cost of and effort needed to reach 20,000 people by foot in Bangladesh in 2 weeks. That would be really expensive. But obviously, we don't know how effective this is compared to the traditional ways of raising awareness. So, but it shows that the potential. So that was the animation project, and there are other projects ongoing now to kind of try and develop this a bit more. But I just wanted to talk about the bigger picture, because when you look at the bigger picture, farmers are the end users of the antibiotics. Like I said before, often they don't actually have very much, often they have reduced or limited choice about their need for antibiotics, because it could be that the water from the environment is polluted, so the high levels of disease. The larvae from the hatcheries might be poor quality. The drugs, they might be getting poor advice or poor quality drugs. So I think it's a lot of the focus goes on farmers, but for long-term sustainable solutions, we need to look at the bigger picture, and there needs to be effort from everybody, because everybody is involved in this. And again, I'd be really interested to hear about your experience in India of looking at the bigger picture, because I think it's very complicated and difficult. So from farmers, obviously, they have hatcheries that supply the larvae. There's the pharmaceutical agents and the feed agents who play an important role in directing what sort of antibiotics are being used. And obviously there's farm supply shops. In Bangladesh, often the farm supply shops also don't have any training, even though they're the ones often providing the diagnostics or advice at least, and supplying the drugs. They don't have any training themselves. And the government does provide training where possible, but is very limited resources. So it's a very complicated picture, and farmers are only one part of this. So the last two projects I was going to talk about, obviously I talked about the animation project. The first one is our project on improving biosecurity in hatcheries. So by improving biosecurity in hatcheries, the idea is that the larvae or the seed, whichever terminology you want to use, received by farms are healthier, better quality, which helps the farmers and hopefully therefore reduces the need for antibiotics. It also hopefully reduces the need for antibiotic usage in hatcheries if they've improved their biosecurity. And the second project is looking at the pharmaceutical value chain. So where are the antibiotics and the other pharmaceuticals coming from to the farmers, and how can we understand this and try and develop interventions to improve the situation and promote more prudent usage. So firstly, improving hatchery biosecurity. So in Bangladesh, there are 60 hatcheries that supply around 150,000 farms. So you can see that if we can improve biosecurity and reduce perhaps the level of resistant pathogens, resistant microbes in hatcheries, we might help to stop the spread of resistance and improve biosecurity across the sector. So this is why we're targeting hatcheries. Also because we know that biosecurity in hatcheries is a huge problem in terms of production and productivity of the industry. The first way we're doing this is through providing bespoke feedback to specific hatcheries. So we're collecting biological data. So we're looking at which pathogens, which microbes that cause disease are present at the different stages of the hatchery process, so we can identify where the biosecurity risks are. We're looking at chemical parameters in the water, like salinity, pH, and water quality, or the water quality parameters. And then we're also collecting socioeconomic data on management practices. And this is important because we need to understand what the management practices are, and so that we can help people to improve these. And it has to be improved in a cost-effective way, because otherwise it's just not going to be taken up. The other part of this project is we're producing digital training materials. So one of the things that everybody is really telling us is that they want more training on biosecurity. So we're producing a set of 7 videos that cover different aspects of biosecurity. This is a recent video of filming on site. And so we're going to, and the aim of these videos is that we are providing the senior hatchery staff with training materials to train their junior staff. So we're supporting the senior staff really, and then hopefully that will trickle down and improve biosecurity in that way. So yeah, the first thing is the digital training materials. The second thing is we're producing a self-assessment tool. So the idea, this obviously won't be in English, it will be in Bengali. The idea is that the senior hatchery staff and the junior hatchery staff, if they can use it, will have an app on their phone that they can use to assess their various different aspects of their hatchery biosecurity. And they can look at it and look at the different green, yellow and red factors and say, okay, well, this is I'm green in this aspect, or they can see that their biosecurity is poor in different aspects because it's red, and they can easily look at overall how their biosecurity is, and they can then look at how they could address this. And this is important because currently they don't have advice very easy to access on how they can improve their biosecurity themselves. And sometimes when it's a more formal assessment process, sometimes they're not as honest as they might be because they're trying, they're being formally assessed. So this allows them to informally self-assess, and they can take advice. And there will also be links where they can get more information if they need to. And then the final project I'm going to talk very quickly about is the pharmaceutical value chain. So the idea is that if we can improve, well, let me go to the next one. If we can understand the pharmaceutical value chain, so where are the pharmaceuticals coming from, who is involved in this, and what are the drivers for all the different stakeholders? Why are they supplying particular drugs, or why are they behaving the way they are? What things do they value in this whole process? Because we want to develop interventions that are going to work long term, and in order to do that, you have to make sure that you're meeting all the stakeholder needs, and ideally all the stakeholders will be involved and engaged, because otherwise, if you develop an intervention that just involves farmers and doesn't involve, say, pharmaceutical company agents, then it might not work long term because you're not going to change the behaviour of everybody. So we've got a number of farm shop owners who have been fantastic and helping us with our understanding of the whole value chain, and they've been absolutely amazing. This is a research assistant, Bashar, who is working with Bangladesh Agricultural University, Professor Mahfujul Haq, and him and a couple of other research assistants have been started off by interviewing shop owners to try and understand the situation, and they also interviewed some pharmaceutical agents and government officials and farmers. And this is very preliminary, so it's not finished by any means. But the first thing we've done is develop a stakeholder map. So you can see that the nodes, so the circles, show how many interactions people have with each other. So shop owners have the most interactions with everybody in terms of pharmaceutical usage and sales. And so they're the main stakeholder involved in all of this. And the thickness of the line shows how many interactions they have with the different other stakeholders. So their main people they work with are fish farmers. This is in the finfish industry, but obviously we could do something very similar in the shrimp industry. But yeah, and so first of all, we're developing this stakeholder map, and then the next stage from here, we held some focus. Well, I say we. This is our in-country partners, Bangladesh Agricultural University. They've been holding some focus group discussions at different upper zillas, different districts. And at these discussions, we have tables of, these are the table of farmers, and we also have tables with farm shop owners and agents and government officials. And we're really trying to now understand the needs and the perspectives of the different stakeholders, and looking at how the different stakeholders perceive this. So, for example, one of the activities we've had them do is a problem tree analysis, where they write the problem on the trunk, and the roots are the causes of the problem, and the branches are the effects of the problem. And this is a really nice way of seeing how, for example, the farmers group sees the problem and sees what the causes are compared to the other groups. And we can start getting a really nice idea of how everybody approaches this. And then we're also doing other activities, and this is one where we ask this is again the farmer group. This is where they drew these sort of stakeholder maps themselves, and then these piles of chips are representing the level of power and influence that they feel that each of the different stakeholder groups has. So we can get a really good idea of how people perceive each other, and also we monitor things like how much money flow of money and the flow of training and resources, all these sort of things, really to get a great idea of the situation. And from that, we'll start developing some potential interventions to test out, and that's the next stage. Then one of the things that has been identified is that there's a real need for more training materials. And so we will also be providing some digital training materials on prudent pharmaceutical usage. So that's another part of this project. And that's it. And so just to conclude, the messages that I really wanted to get across, I hope I have, is that aquaculture does pose an important risk to AMR, and that's global AMR, and through a variety of ways. We need to find ways of reducing the use of antibiotics in aquaculture, and this requires working with all stakeholders, not just farmers, if we want long-term impacts. But obviously farmers are important as well. We need to find solutions that work across the sector. Okay, and so thank you all for listening, and just say thanks to, in particular, these collaborators and all the people I work with and the funders.
Thank you, Dr. Kelly. It was very informative, and thank you once again for sharing your experience. This will be very useful. So, Dr. Satisha, is there any questions for Dr. Kelly?
It was very informative presentation. Thank you. So during this conversation, just I want to brief, like Dr. Kelly has explained what are the different type of antimicrobials are used not only for aquaculture but for medicine as well, or even oral food sector as well. So then how this AMR issues are coming up, what is the issues, and how we can have to come out to solve this problem, what already has started, and how we can come out. So Madam has already worked for several years in Bangladesh and very good experience, and being very, since India is sharing water with Bangladesh, quite similar geographical condition, though aquaculture practice quite different here. So, but since the environmental conditions are quite same, so probably the results might be having some kind of similarity. But some questions just wanted to know, like what kind of antibiotics they were using in Bangladesh, like OTC or this internationally approved antibiotics, or even.
Yeah, well, so it's difficult to get this information, obviously, because like I said before, I think OTC is the only one that's approved for aquaculture usage, and that is used. But from some of the data we've collected, there are unapproved drugs being used quite extensively.
Because if we go with all the usage pattern, the quantity used or marketed, and how it is coming to that, because this is a highly unorganized sector, probably, yeah, it's quite complex, but maybe very difficult to correlate between this usage and the practice in the aquaculture.
Yeah, I think it is really difficult. I think, first of all, getting accurate data on usage. So we have some partners who know the farmers really well and have earned their trust, and they're a bit more honest. And some of the, like ciprofloxacin is often being used. Drugs that really shouldn't be used in aquaculture. But also, like I say, sometimes the residues might be coming from elsewhere, so from the water entering the farms, But one of the things that we'd really like to do in the future is somehow to try and measure or try and correlate the resistance and the antibiotics that are being used. So that's one of the projects we're doing at the moment, is looking at, we've taken samples from ponds and we've DNA sequenced them, metagenome sequencing, which means sequencing everything that's there, looking for AMR genes to see which resistance genes, resistance to which antibiotics are present, and does that correlate with the antibiotics that we think are being used? So that's one of the things.
Yeah, but now issues like now if we are doing like a AMR bacterial survey in an aquaculture system, there's definitely, because of this complexity of farming system, possibility is very high that we'll be getting some bacteria with the resistance. However, we do not know what is the real cause, whether the bacteria itself has come from outside because of some other contaminated sources, or it has because of the entry of antibiotics or this antimicrobial entry into the aquaculture system, since water is a universal solution. So how to tackle this issue? Like the source of antibiotics or antimicrobials may be domestic sewage or industrial effluent, but aquaculture is taking the blame.
Yeah. I mean,
how we can address this issue?
Yeah, I think it's really important question, and that's why I was saying I think we mustn't just look at farmers and aquaculture at farmers as the end users. It's a bit like human antibiotic usage. All the focus is going on individual patients, when actually, we need to look at society more generally. So for aquaculture, without extensive sanitation infrastructure, wastewater infrastructure, it's not sustainable because the farmers don't have so much choice. So I completely agree with you. The other thing I was going to say was, so one of the projects, our hatchery project, what we're actually doing is we've taken samples from all of the inputs, and so we're looking for AMR resistance genes and pathogens in all of the different inputs, and then in the larval rearing tank and the different tanks, to try and maybe not identify where the resistance genes are coming from, but where the pathogens are from. And by looking at that, we can say, well, actually, we can see that the biggest source of pathogens is the broodstock, or it is the water, and at least that gives us the start of an idea of where the biosecurity risks are. But you're right, it is a really complex problem.
Yeah, because like a shrimp aquaculture, for example, it's more of commercial practice, but a fish culture like carps and all, they are more of livelihood. The margin what farmers will be getting is very minimum. So establishing huge biosecurity measures or improving the management practice, it will be very highly cost involving. So how much, whether they can sustain economically, that's a big question.
Yeah,
and
yeah.
We have to come out with some solution. Yeah.
Exactly, and I think that's why, I mean, it's difficult for me because obviously you guys understand the local context a lot more than I do because I'm based in the UK, but I think that for me, that's why it's really important to work with the stakeholders really closely and really get a good understanding of what their practices are and what are the potential solutions, because like you say, there might not be huge solutions, changing wastewater infrastructure. I mean, that is billions and billions of dollars of, but maybe there are small practices like in our biosecurity, hatchery biosecurity. Already we've identified small practices that people could do that would just improve things generally, like just making sure that no visitors are allowed that have been in, anywhere near shrimp for 72 hours before, or having better practice inside the hatchery. So I think that's a bit different, but I think by working with stakeholders we can try and identify some solutions.
Yeah, thank you. That it should be a holistic approach than one sector. Yeah, thank you. Thank you very much. So shall we continue?
Okay, so thank you, Dr. Kelly, once again for the very informative talk. and we'll move to our next speaker. So Dr. Anusa Rohit. So she's heading the microbiology division in Madras Medical Mission in Chennai, India. She's also the chair of infection control. She has been continuously speaking in many forums, and we are really fortunate to have you with us. So now I request Anusa to deliver her talk.
At the outset, thank you so much, Dr. Ota. Know you since being a very small girl, hardly into AMR, but now AMR has become the talk of the town. So we will have to probably learn to be a part of the whole spectrum. So without further ado, I think I will share my slides so that So my talk is going to be completely on antimicrobial resistance from the clinical point of view, because as you all know, I'm a clinical microbiologist. So for me, it is very important that we are able to look at AMR on an individual point, as well as from infection control on a bigger spectrum. So going right over, I bring you greetings from the Madras Medical Mission Hospital. And why do we need to learn about resistance? So is it just about, talking about each of us? Now, for example, the question asked with the previous speaker was, why is it that the farmers need to know about antimicrobial resistance? Now, this is precisely, I think, why we need to talk about it on common fora, because each of us is talking individually in our groups, but what we need is a combination of all of us working together to look at reducing antimicrobial resistance in the world, rather than saying we will only reduce it among the aquaculture industry, or we will only reduce usage among the healthcare industry or the pharmaceutical industry. I don't think one methodology is going to help every aspect to reduce this problem, which is probably greater than the COVID-19 crisis, but we seem to be worried more about COVID-19 than we are worried about antimicrobial resistance. So high time we talk about this. So no better time to talk about it than the World Antibiotic Awareness Week. So this is really like a silent tsunami that we are facing. It is having numerous health and political summits that have been speaking about it. There's a great deal of data that exists in the form of various reports, but how are we really taking it back into the community? Like, for example, you may all be aware that clinically we had all given up, Dr. Abdul Ghafoor and team from Apollo Hospitals had given the Chennai Declaration, which came out almost 7 to 8 years ago, but everything's on paper, nothing's really coming on ground. And we still face a huge problem with MDR gram negatives with each passing day. So if we look at how antibiotics appeared across the years, and you see on the other side how resistance organisms were identified, you will see that there hasn't been too much of a time lag between the introduction of an antibiotic in the community and for the resistance to develop for these antibiotics. So we are talking about penicillin being introduced in 1943, and by 1944-45, we were already having penicillin resistance. So if you look at the timeline here, again, very alarming to see that we have organisms with huge amount of resistance to tetracyclines, to macrolides, to gentamicin like aminoglycosides, even to glycopeptides like vancomycin, and to carbapenems, to right now we being at a really dangerous stage when we have not too many new antibiotics coming up, but we already have almost come to the end of the tunnel as we are looking at, with no light in sight with new antibiotics and antimicrobial resistance. So if you look at all the antibiotics that are acting on the various parts of the cell, bacterial cell, you will see that antimicrobial resistance is is part of each of these groups and as a whole. So the former WHO Director General had said a post-antibiotic era means an effective and an end to modern medicine as we know it, and things as common as strep throat or a scratched knee could kill once again. Now, we've never been able to accept that. So antimicrobial resistance is when germs, that is bacteria and fungi, or even viruses right now, develop the ability to defeat antibiotics designed to kill them. It does not mean your body is resistant to the antibiotics. So, of course, this was previously already explained by the speaker, but I'm going to go through it once again. So this is how resistance happens in nature, that you have a lot of germs and then you give a low dose or an inadequate dose or an inadequate course or an exposure to an antibiotic, and you end up killing all the good bacteria, and the drug resistant bacteria can now take over and multiply. And the additional advantage is the selective advantages that bacteria can carry resistance mechanisms and transfer it across various other species is compounding the problem. So examples of how antimicrobial resistance spreads and why the community is equally important, in a role to play with this is you will see here that, healthcare facilities cannot act on an individual basis. We are all our industries, whether be it veterinary industry, be it aquaculture industry, be it agricultural industry, ultimately it goes through the food chain. And here you will see that there is a particular patient. He gets an antibiotic and he develops an antibiotic resistant bug. The animal, he is able to give out this antibiotic resistant bug, which is exposed to various animals. Now, the drug-resistant bacteria remains in the meat of these particular animals. The fertilizer or the water containing the animal feces with the drug-resistant bacteria goes into the water stream or into the food chain. And drug-resistant bacteria from animal feces can be explained, can remain on the crops, and then when we eat it, we are consuming it. Now, when we are having these kind of bugs, and you get into a healthcare facility for basic treatment, you end up developing resistant infections because your gut is already used to a colonization with resistant bacteria, and hence you are already coming with a negative a negative part of your gut is already there when you're going in for treatment, which gives you definitely a disadvantage. So germs or bacteria or fungi are everywhere, and they can help us. Some of them help people, get better, and some help them get sick. So when you have a resistant bacteria and antibiotics kill these germs, again, you will see in the same picture that you can multiply, and once resistance emerges, it spreads across various countries. So if you look at whether the aquaculture industry, because I was looking when I was seeing the target audience for this, that we had to look at it from the aquaculture industry. So I looked at a couple of papers from my mom and dad's group, and I found that they are also having drug-resistant bugs that they are identifying in organisms like Vibrio vulnificus. And other than the virulence mechanisms, there's already resistance mechanisms that these are not common organisms, yet they are carrying these antibiotic resistance bugs. So what is the impact on current and future patients? So there are two things that we have to look at. So for most medicines, side effects are limited to the individual patients. Whereas when resistance develops from exposure to an antibiotic, it will affect not just that patient, but future patients, because once you are in the hospital and your gut has the resistant bug, now when you are able to contaminate the hospital environment, the hospital then is teeming with resistant bugs, which are then transferred to susceptible patients. And then that is how antimicrobial resistance in the community increases. So this reduces the number of effective antibiotics we have to treat. So if we look at across the years from 2000 to 2015, if you look at the data from India, if you look at narrow spectrum penicillins, which are essentially resistant, there's hardly any usage at all. Whereas if you look at fluoroquinolones there has been a increase and then a decrease, and now it's attained a plateau. Look at the macrolides, there's a steady increase. Trimethoprim is a steady usage. Cephalosporins went in the early 2000s from literally very few DDDs, that is daily defined doses to be used per 1,000 population, to becoming one of the most commonly used antibiotics. And then came broad spectrum penicillins like carbapenems, and then tetracyclines also, which have been used extensively. So there are intrinsic resistance mechanisms that these organisms are inbuilt with. And then there are acquired resistance mechanisms that we should look at. So why should we really worry? So the WHO and the CDC have given urgent threats, emergency that we need to look at. So carbapenem resistant Acinetobacters, which especially are isolated from ICUs, Candida auris, which is pan drug resistant, Clostridioides difficile, which is resistant to most of the antibiotics, carbapenem resistant Enterobacteriaceae and other pseudomonas, and carbapenem and Neisseria, which is resistant to many of the antibiotics, are big problems. The next problems are the serious threats that we have to look at, for example, drug-resistant Campylobacter, drug-resistant Candida, extended spectrum beta-lactamase producing Enterobacteriaceae, vancomycin-resistant Enterococci, multi-drug resistant Pseudomonas aeruginosa, drug-resistant non-typhoidal Salmonella and Salmonella typhi, drug-resistant Shigella, MRSA, drug-resistant strep pneumoniae, and drug-resistant TB. Now, if you think that these are all names that are only coming in these reports, let me be very, very honest with you. This is what we look at on a daily basis in a clinical microbiology lab. We have patients coming in from the community. Even this morning, I reported a urinary tract infection in a patient which was pan drug resistant, including to colistin and fosfomycin, which were our only options for choice of treatment. Concerning threats are the erythromycin-resistant group A strep and the clindamycin-resistant group B strep, and watch out for the azole-resistant Aspergillus fumigatus, Mycoplasma, and Bordetella. So, coming back to what are these, I'm going to deal with the gram-negatives and the most common, antibiotics that we are looking at that are resistant. So, what do you mean by ESBLs? ESBLs stand for extended spectrum beta-lactamases. And these are enzymes which confer an organism resistant to first, second, and third generation cephalosporins, and also Astronam. And you have 4 classes depending on how they are different enzymes. So it is a group of over 150 to 200 enzymes in each group, which essentially confer resistance. They may differ from each other with only a single amino acid, but they are a different enzyme altogether. And they have differences in what can be used as inhibitors for them. So there are, most of the ESBLs are inhibited by clavulanic acid, but there is an AMP-C producer, especially with Enterobacter and certain Klebsiella, which are not inhibited by clavulanic acid as well. So as part of my PhD post-MD, I looked at about 300 isolates of ESBLs from the Madras Medical Mission, of which 88% were CTXM types and 12% were non-CTXM types. All 300 of them were chosen because they were ESBL producers. The CTXM types were further grouped, and we looked at and found that quite a few of them carry more than one type of enzyme responsible for resistance. Now, this is a table which classifies class A, class B, class C, and class D type of carbapenem of ESBLs, and you will look at and see that there are so many enzymes and so many inhibitors that actually come into being. Now, why is this mechanism so important, worth studying, is that the emergence of different CTXM type of enzymes was first introduced in 1990 as a concept. Between 1994 and 2000, we saw certain successful enzymes that were isolated epidemiologically more and more. And after 2000, there's almost been a universal dispersion and globalization of this enzyme. Now, this enzyme is also actually on the plasmid, so it is transferable with other organisms. It is also flanked by transposons, integrons, and other insertion sequences, which makes it a very complex yet a very mobile genetic element which helps in spread. So what do you mean by these mobile genetic elements? So they can either spread through a bacteriophage, which is called transduction, or by conjugation, that is, it can be transferred between germs through a sex pili or through transformation, and that is again very, very important. Now, if you look at the different kind of CTXMs that have been in Europe, you see pre-2005 and post-2005, you will see the spread has been successively increasing. You will also see this here again, and this is coming to a single isolate that we looked at, which had two types of ESBL genes that we were able to recognize in our sequence. It carried a BLA CTXM 15 in the plasmid and a BLA CTXM 27 in the chromosome. So what it also shows is that this enzyme is so mobile within the cell, bacterial cell, and it is able to also maintain these enzymes in the chromosome for posterity. Now, not only were these organisms, also carrying these CTXM kind of genes, they carried resistance to multiple other antibiotics, and hence we are actually in a very, very grave situation with our inability to use any cephalosporin for treatment in patients. And it penetrates the landscape and is present in the veterinary and the agricultural industry as well. So CTXM has been found in food, pets, in wild animals, and in river water. And these enzymes have been isolated in Enterobacteriaceae and non-fermenters. Large number of enzymes are grouped into one family because of their genetic similarity. And it is important to remember that each industry does contribute to this. So when you have a patient coming into a hospital with a community-acquired UTI, which is resistant to antibiotics, it is not a simple job to treat the patient. It may be talking about one patient, but for that patient, we are stuck with using an IV antibiotic that requires hospitalization, increased costs, increased chances of morbidity, other mortality, etc. is a definite problem. Now, the second group I'm going to talk to you about is on carbapenems. Carbapenems are like our ICU, our AK-47s in the ICU. This is one antibiotic that helps penetrate the bacterial cell wall and is able to bind to enzymes known as penicillin binding proteins. So it has a lethal effect, and it's very good because this group of antibiotics are active against gram positives, gram negatives, as well as anaerobes. Okay, and it is very, very good to treat patients. But transmission of different resistance has also been seen in soil bacteria, and carriage of such genes was literally exchanged to clinical pathogens, and multiple mobilization sequences, including non-coding regions, were identified in short region sequencing. Now, this is essentially why the NDM metallobeta-lactamase, which was first reported from Chennai, was from sewage water. And these are mobile genetic elements that have been found in Enterobacteriaceae in the environment. And when it affects a patient, essentially you are only looking at antibiotics like, colistin, which are very toxic to the kidney. They have a lot of side effects. You are also looking at the end of the road as far as treatment of many infections. You may use newer antibiotics like ceftazidime, avibactam, etc. Please remember, even a carbapenem like meropenem, you are talking about treatment of rupees 8,000 to 10,000 per day for the antibiotic only. And this kind of a patient is generally in the ICU with extremely high costs. And that is a per day requirement for antibiotic for at least 8 to 10 days. So the antibiotic cost alone is going to end up leaving the patient penniless. So this is not really something that we can really think as, that we don't need to worry about. It's not in my environment, so why do I have to really think about it? So going right into the actions, there are different mechanisms of resistance among these groups of antibiotics as well. But what is most important is what we are seeing from the Indian consensus in terms of the kind of carbapenemases. So NDMs along with OXA48 is what has been seen. A point of care tool is very, very important in diagnosing early, and strict contact isolation and hand hygiene are crucial in the prevention and treatment of ESB, of carbapenemases in the hospital. Now, it also makes when an antibiotic, when a pathogen is resistant to even carbapenems, you are almost rest assured that this organism is an ESBL producer. You're almost rest assured that this organism may carry fluoroquinolone resistance. You may or may not be seeing aminoglycoside resistance, but essentially it also means that you probably have one or two choices of antibiotics that you can possibly choose. And here it may be a compromised patient with having a kidney failure already, and we may not have the opportunity to be able to treat this patient with this kind of an antibiotic. So the WHO also released the list of the world's most dangerous superbugs, and all the organisms that I spoke about are already part of this group. Now, where do these organisms stay? For example, let's look at the habitats of these various organisms. So we are talking about Klebsiella, the natural habitat being the GI and the respiratory tracts of animals and humans, fruits and vegetables, dairy products, wood pulp, paper mills, water storage tanks, surfaces of water, sinks and drainages, etc. So are we in contact with any of these? Yes. Okay. And this is an organism which is really difficult to treat. Trust me, it is probably the biggest burden that we will be facing in the world tomorrow. Next, we're talking about Enterobacteriaceae, again, very common in the gut of various organisms and also in the raw meat, in soil, in water, in sewage, in sink drainages, etc. Acinetobacter is found on the skin and mucous membranes of pets and animals, in shrimp and water farms, fish farms, sewage, dump sites, water treatment plants, sinks and drainages, petroleum hydrocarbons, soil, warm and humid temperatures. All of these are able to make the organism survive. And then you have Pseudomonas, which is again present in human skin, upper respiratory tract, GI tract, raw milk, moist environments, including the soil. So now inside the hospital, look at this scenario. The patient is colonized with a multi-drug resistant organism. Now this may form biofilms on various surfaces, like for example, the hand wash basins, the toilet rims. And then the patient goes, say, for a colonoscopy, and the colonoscopy or the endoscopy uses, a particular scope that you put into the body. And this has been, high-level disinfection has been done with, say, glutaraldehyde or an OPA, but this organism is multi-drug resistant and may also be resistant to these biocides, which essentially helps it transfer from one patient to the other. Hospital linen, keyboards, high-touch surfaces like doorknobs, switches, etc. are other places where these organisms spread. So if you look at India, we're actually right at the top with the amount of carbapenem resistance that we are seeing. And the susceptibility in different isolates in our hospitals has reduced from 80 to 68%. So do the drugs work less? Probably, yeah, because we are sitting on a position where we are looking at extreme drug resistance. And if you look at NDMs in India, we are again at the top. So we are champions with a lot of things, including ESBLs and carbapenems. So these are all the unique beta-lactamases that have been identified. So rising income and increasing access to antibiotics are saving lives for sure, but they're not a good substitute for public health. So what do we need to do? Good infection prevention and control in hospitals. Tracking and sharing data. Unless we all sit and talk on the same platform, we're not going to get much further. Antibiotic use and access, vaccines, therapeutics and diagnostics, environmental sanitation, and the clean environment that we stay in. These are probably going to really help. because access to safe water has literally become nil. There is a beautiful documentary on Netflix on the, on how fresh water is going to almost become, something that we cannot have access to within a couple of years world over. So if you look at this graph here, you will see that, there is a sanitation access on the left side and the ESBL prevalence on the right side, and India being right on top there. You will see that wherever there is, sanitation access is very poor, the chances of ESBLs are extremely high. So antibiotic consumption is also increasing because obviously we are treating more and more resistant bugs. So we have to go for higher and higher antibiotics. And as you see with time, we've been increasing the use of antibiotics in the healthcare system. But trust me, here we have no control on being able to, not use an antibiotic. There are small measures that we can put in place. But the bigger problem is the patient coming in with multi-drug resistant organisms and the general access to safe water being very, very poor. So total antibiotic consumption, you will see we've gone like way up between 2000 and 2010. You will see the difference has been phenomenally high, and carbapenems also we've been using extremely high. And I think this is something we need to understand, that there is a high mortality and a high potential for spread that we are looking at. So gram-negatives really contribute to the bigger problems in the ICUs, and multi-drug resistance is noted in almost 87. So you're talking about 90% of the Acinetobacter, 75 to 80% of Klebsiella, 60% of E. coli, and 58 to 59% of Pseudomonas in the ICUs, and they are responsible for hospital-associated central line-associated bloodstream infections or catheter-associated urinary tract infections. These are our big problems, along with ventilator-associated pneumonias. So shall we just wait for the government to give us guidelines, or shall we sit together and talk? And that is where I think we all come in. Resistance trends can always be very local. So these resistance trends may vary between place to place, between regions in the country, between hospital units, and between pathogens. Like, for example, in our hospital, Klebsiella is our biggest problem. Acinetobacter is not. So there are probably reasons why this organism is more commonly here. So there is a global availability of colistin, which is like an OTC available in this country. And as you know, colistin resistance has been spoken about, especially from China. The poultry industry has been speaking about it very commonly. It is plasmid-mediated MCR1. Luckily, in India, we're seeing less and less of plasmid-mediated colistin resistance in colistin. Most of them are MGR1s, which are MGRB gene related, which are chromosomal in nature, which is why probably it still hasn't become as rampant as what we would expect in a condition like India, where we have to use colistin in most of the places. So countries reporting plasmid resistance to colistin, we are probably here, we don't see it much in the animal industry, nor in the environment. We are looking at it more from the hospital data. So what can you do? Important to start looking at that. Use antibiotics when prescribed by a healthcare professional only. Always take the full prescription. Never use leftover antibiotics. Never share antibiotics, and prevent infections by regularly washing your hands, avoiding contact with sick people, and keeping up to your vaccination. The other preventive strategies include curtailing production, consumption, especially in human and veterinary medicine and in agriculture, education of the general population and the healthcare personnel, veterinarians and pharmacists about the means for prevention of proper usage of antibiotics. Offering access to clean, affordable water and sanitation to all people, and promoting vaccination and preventive health, introducing animal breeding and food production processes that render the use of antibiotics unnecessary. So this kind of a program needs us to work in tandem and not as two separate streams. So implementing a mandatory antibiotic stewardship policy is most hospitals' primary outcome now. We are also doing extensive surveillance, recording, and reporting. We are part of the WHO WHONet network that we are entering our data into to analyze our data. Patient history and risk-based screening is also very, very important. Assessing medical and hospital stay in the last 12 months, that that is important history for the patient, and training and education of the personnel for screening and contact precautions are also what we can look at. So first is that we have a good diagnostic technique for the hospital. We need standard infection control protocols. We need additional protocols when we are dealing with MDR, and decontamination processes and cleaning processes are very, very important. So it's important that you are part of this problem that we are facing, along with your loved ones when you are coming to hospitals too. And antimicrobial resistance is a worldwide global problem. It is not restricted to the healthcare industry. It is not restricted to the veterinary industry. It is not restricted to the pharmaceutical industry. It is restricted to nowhere but everyone. And I think what we need to do is work together. With that, I'd like to conclude. Thank you so much for the opportunity.
Thank you, Dr. Anusha. It was a wonderful talk, and you have had a wide coverage regarding the problem and how to take precautions, all these aspects. So I'll request Vidya if any questions is there.
There is no questions, sir, but we have some inquiries regarding the effectivity of our healthcare system in tackling this antimicrobial resistance. How much this is giving to the common people, which is in the grassroots level, ma'am, and what is the agenda of Indian government to tackle antimicrobial resistance? Is there any programs they are organizing or they are going to organize? Kindly enlighten us, ma'am.
Thank you so much. So definitely we are doing a lot of surveillance, and we are seeing, for example, among gram negatives, ESBLs are at about 80%, even from the community. We are also having a lot of NDMs from the community. We're talking about 20 to 30%, which can snowball to become a huge percentage if we are not careful. Colistin resistance is still at about 1 or 2%, but we are not far from misusing it. And we are looking at resistance mechanisms that are across the spectrum for community acquired infections as well. So that's answer to your problem one. The second question is, yes, the government is doing, but isn't it our individual, each of us are working as individuals. And I think that is where the biggest gap remains in the system. So the government creates policies, the antibiotic policies are there. There are so many programs this week on the World Antibiotic Day, Awareness Week, from the government as well as from individuals and institutions. But I think it's just not enough. We need to work as a team together, as a forum where we can address this.
Yes, ma'am. And ma'am, one more question, ma'am. If a patient is admitted in a hospital and he had a surgery, after that he has been getting if it is seen that it is occurred from the hospital, any claim the person can make with that? That is the another question, ma'am.
Sorry, I didn't get the question. You were broken. Would you mind repeating the question?
Yes, ma'am. One person is had a surgery in a hospital. So
Your voice is breaking, Vidya.
Who gets to pay? Is that your question? Who gets to pay? Is that your question?
Yes, ma'am. If it is hospital borne, the superbug infection is hospital borne.
If the antimicrobial resistance came from a fishery industry, does the fishery industry pay? It does not work like that. We cannot be, we are not, at least our industry is not insurance based like how the US is. And that is why the individual patient only pays for every infection that happens. The burden is on the patient. If the microbial resistance, if a patient came in with a multi-drug resistant bug from the community, who would pay for it? We cannot be pinning the blame.
Hello? Hello? Dr. Anusha, are you online?
I can hear you if you can hear me. It's Kelly, but I can't hear anybody else.
Dr. Anusha? Patil sir? I can't hear. You're unmuted.
Yeah, I'm sorry.
It's okay. So any other questions, Vidya?
No, sir. It's enough.
Okay. Thank you. So thank you, Dr. Anusha. Thank you so much.
Thanks.
So we'll move to our other programs. Now I request Dr. Alavandi sir to speak on this occasion. Alavandi sir.
Hello, good evening.
Yes,
Am I audible?
yes, sir.
Oh, so good afternoon, everybody. It's a nice afternoon, but we have a lot of rains here. And nevertheless, the program has gone on well. I'm very much thankful to our international speakers, Dr. Kelly from University of Exeter and Dr. Anusha Rohit from CMC Chennai. I know earlier I could not catch up with the talk by Dr. Kelly. I hope our people enjoyed her talk. And one point I just picked up from Dr. Anusha's last part, that is the responsibility of each and every one of us. The basically we all have the rules and regulations in place. Government is enacting all kinds of rules and making us responsible. But finally, the responsibility lies with the end users, basically the people. So this is where especially we in the developing countries are it's very important that we follow the prescriptions given by the doctors and all that. So that is one aspect. But the antimicrobial resistance problem has been time immemorial, and actually, if I just recollect, I started my career way back in 1985. Since then, we have been talking about antimicrobial resistance. How do we overcome? Then, the problem started aggravating, like the methicillin-resistant Staph aureus infections, and infections of multi-drug resistance Pseudomonas, and then infections due to the drug-resistant Mycobacterium tuberculosis. So several of these things have been, still serious challenge to the human medicine and also in the veterinary medicine. But when we come to the environmental this thing, I think we still don't have much of the serious problem with regard to use of antimicrobials here. But nevertheless, it's in some of the publications are showing that the use of microbials is increasing the aquaculture sector. But I'm very happy that for the benefit of our two speakers, last year our people conducted a research program on the use of antimicrobials, and actually when the antimicrobials are found in the shrimp or the seafood what is exported overseas, it is actually if they catch any presence of antimicrobials in the consignment, they just reject it. But if the study conducted by our people for the last 15 years has indicated that the use of antimicrobials is significantly reducing, and especially we are now taking lessons from countries like Norway and Scandinavian countries where the salmon farming, they used to use hundreds of tons of antibiotics, and now most of their diseases they are controlling using vaccines. And in the fisheries and aquaculture sector also, we have initiated programs on developing vaccines and other preventive programs. And we ourselves at SIBA, we have recently come up with bacteriophage mediated control of infections due to bacteria in the aquatic systems. So there is definitely progress happening. And in addition to this, I just wanted to mention that this problem of antimicrobial resistance is time immemorial, as I said in the initial few minutes. Permafrost soils or something. Years old. We need to really think whether this is this problem is new, or how can we tackle it, or have we necessarily because one definite problem is that we don't have enough options for treating the especially the humans when they we are stuck with multi-drug resistant pathogens. So we definitely need to be concerned, and we need to work on more alternative approaches. Like Georgia, they rely on the bacteriophage therapies, especially with regard to treatment of enteric infections, and also cases like diabetic foot infections, bacterial infections are controlled by use of bacteriophages. So such options we need to really pursue, and so that's what I just want to highlight in this on this occasion. I know that many of our children of our colleagues were involved in understanding what are antimicrobials, how they're used, how they're misused, all those kinds. And I'm very thankful to all my colleagues that they have been successful in educating their children that how judiciously we should use the antimicrobials in our systems, and such education is very important, especially we the elite circle, educated people. We should educate our own children and nearby ones, so that at least that is a beginning. We know that we have a large population. We still need to educate them. Basic education is required, and in addition to that, we also need to impart good practices, including these important aspects like antimicrobials, use of antimicrobials, responsible use of antimicrobials. So with this, I congratulate the team who have been involved in creating awareness, at least among the SIBA fraternity. This information, I hope most of the children also will remember. Anyway, once again, before I close, I thank the speakers, Dr. Kelly and Dr. Anusha, and all my colleagues who have taken up this venture. Thank you very much.
Thank you, sir, very much for your wonderful interpretation while analyzing the different situations and also providing a number of suggestions, and definitely it will educate the people. And now I request Dr. Bhuvaneshwari to move to the next our step of the function, announcement of awards. Bhuvaneshwari, please.
Good afternoon to all. On the occasion of World Antimicrobial Awareness Week celebration, competitions for research scholars of our institute and school children of SIBA staff were conducted to create awareness. The essay competition on the role of citizens in making India safe from antimicrobials was conducted on 18th November. The first prize winner is Mrs. Radhika. The second prize winner is Mr. Sudama Swain. And third prize winner is Miss V.R. Vandana. The elocution competition on topic Can We Avoid Antimicrobials for a Safety Society held on 19th November. The first prize goes to Miss V.R. Vandana. Second prize is goes to Mr. Dibin George, and third prize goes to Mr. A. Suresh. The drawing competition for school children was conducted on the topic Microbials Handled with Care. Now I will upload the PowerPoint. Can you see, sir?
Yeah.
Based on their theme, creativity, color combination and neatness, the first prize was shared by two Ananda Jaradan, son of Dr. Ananda Raja, and Eric Fenas, son of Mrs. Prasanna. The second prize was also shared by two Aishwarya Durga, daughter of Mrs. Madrabhutu Bala, and by Pavan Kumar, daughter of Dr. Geeta. The third prize was shared by three persons, Pragya Anand Singh, daughter of Dr. Shine and Sujit Kumar, Ayushi Ghoshal, daughter of Dr. Ghoshal, sir, and Shreyas Das, son of Dr. Sanjay Das. All the participants will be provided with a certificate of participation. I thank all the participants and parents of all who participated for their efforts in making this event successful in creating awareness to young minds for safe use of antimicrobials to save nature and humankind. Thank you.
Thank you, Dr. Bhuvaneshwari. So now I request Dr. L.L. Praveena to propose the formal vote of thanks. Unmute, Dr. Praveena, unmute. Yeah. It's breaking.
I extend my sincere gratitude towards the speakers for spending the valuable time and gracing the webinar. I have a deep sense of appreciation to Kelly for sharing her experience with us. Thank you, Dr. Kelly, for invitation and giving an excellent talk on aquaculture. I am also grateful for also creating among us through the webinar. I having us time during the time. Next, Dr. Kelly for his constant support and encouragement to carry out this. I thank our graduations for conduction of this webinar. My sincere appreciation the organizers, Dr. Stakeholder and Dr. P.K. for making necessary arrangements for this webinar and acknowledge the by Dr. Antaja, Dr. Bhuneshwari, Dr. Satish Avanju, Dr. Vidya, Dr. Satish Kumar, Mr. Dr. Vinay, who played role as judges for various competitions held during the AMR Awareness Week. My sincere appreciation to Dr. Mohammed, Dr. Kumar, and Rajan for making arrangements for the smooth conduct of this webinar. Finally, I thank all the participants who have actively participated in this webinar. Without their active participation, this webinar won't be successful. I thank one and all. Thank you all.
Sir, unmute.